Cool. Hello, everyone, and welcome to today's live presentation, Utilization of Stem Cells to Crack Motor Neuron Development and Degeneration. I am David Cunninger from Thermo Fisher Scientific, and I'll be your moderator. Before we begin, I'd like to point out that you can submit questions at any time by typing them into the Q&A box, which can be found in the lower left of the presentation window. We'll answer as many questions as we can following the presentation. I'd now like to introduce today's speaker, Dr. Jun-An Chen. Dr. Chen is an assistant research fellow at the Institute of Molecular Biology, Academia Sinica, in Singapore, where the focus of his research is to elucidate how neurons establish individual identity in the developing nervous system and why only specific neural subtypes are vulnerable in various neurodegenerative diseases. He has developed and generated a series of stem cells in animal models to study the functions of micro and long non-coding RNA in gain-of-function and loss-of-function approaches. Dr. Chen is also a core member of the neuroscience program and RNA program at Academia Sinica, which provides a strong and solid consortium to give advice and exchange of ideas for member projects. I will now turn it over to Dr. Jun-An Chen for his presentation.
Thank you, David, for a very kind introduction, and I would like to thank Thermo for giving me this opportunity to talk about some of the current and future direction of my laboratory. And I particularly would like to thank Tracy for technical help during this presentation. So as David said, my lab is interested in using stem cells to tackle how long non-coding RNAs function during motor neuron development and degeneration. And we know that all of our daily movement and the animal escaping behavior is mediated by the coordination of spinal motor neurons. So in human, in this slide, you can see the exercise, everything is coordinated by the perfect 500 motor neuron subtypes motor circuit output. And if this circuit is wrong, then there's some disease will leading to motor neuron degenerations. And if we look at this, you can see that it's estimated about 500 different motor neuron subtypes exist in human. That is pretty according to the muscle types they are innervating. If we centralize to look at the brachial motor neuron, you can see from the different muscle for our forelimb, and each of the muscle type is innervated by different motor neuron subtype. So the question is how this diversity of motor neuron subtype is established during development. And to tackle this question, we use embryonic stem cell approach as well as mouse genetic models. And we would like to decode how motor neuron diversity is established during development. And furthermore, in today's talk, I will elaborate a little bit more. By using this approach, we can also extend it to see how motor neuron is degenerated in particular disease such as spinal muscular atrophy and amyotrophic lateral sclerosis, which I will focus later in the second part of my talk. Then my lab is interested to see how non-coding RNA is applied in these two subjects. And hopefully we hope this could provide some new therapeutic approach for motor neuron disease based on non-coding RNA. So the first question I would like to discuss with everybody is, can we harness embryonic stem cell into motor neurons? And this is a fascinating question, as the motor neuron development has been one of the best subject and topic of developmental neurobiology. So in this slide, you can see Tom Jessel and James Briscoe have shown very nicely that the development of motor neuron is mediated by extrinsic signal secreting from notochord and the flower plate. So in this case, you can see Sony HR secreting from notochord and flower plate, and it's created a gradient. And this gradient will later on turn on different classes of transcription factors. So these transcription factors usually abut each other. So the ventral neuron progenitor domain will be established. In this slide, you can see motor neuron progenitor is marked by the red color, shown as PNN, and PNN will later give rise to all kinds of 500 different motor neuron subtypes I just mentioned in the beginning. So, and this generic motor neuron will later be divided into the different subtypes according to their position along the rostrocaudal or the anterior-posterior axis of spinal cord. As turned out, the second set of morphogen called retinoic acid and FGF secreting either from the anterior side or the posterior side will turn on different set of Hox gene, and this is a beautiful study by Jeremy Dayson and Tom Jessel, that those Hox genes can define the different subtypes along the axis. For example, in this case, you can see Hox6, Hox9, Hox10 first define the brachial, thoracic, and lumbar spinal motor neuron identity. And in the Hox6 brachial segment, they can further divide it into the Hox5 and Hox8 positive motor pools. And those pools will innervate into the proximal and distal muscles. So this has been a quite nice paradigm to show how motor neuron is generated during development. So Hennig Whitley in Columbia University has shown that this developmental biology textbook study can be applied into the stem cells. So you see in this case, embryos sequentially getting the RA, Sony HR, as I just mentioned, eventually to become post-mitotic motor neuron. So when Hennig was in Tom's lab, he established a beautiful approach that he could apply retinoic acid and Sony HR into a petri dish, and to that the ES cells aggregate as an embryonic body, and this can sequentially be harnessed to become post-mitotic motor neurons, reflecting by the motor neuron specific marker called HB9 in the right panel. So then you might wonder, this doesn't look like a neurons, but because neurons have axons, dendrites, so that's when Hennig later dissociate those embryonic bodies and culture on a petri dish, and he can begin to see motor neuron express a master marker called choline acetyltransferase as motor neuron cholinergic neurons. And if many people and outside of the field did the electrophysiology and co-culture experiment, they can see the action potential is recapitulated in in vitro derived motor neurons. And I think when we and other people co-culture motor neurons with the muscle cells, then you can begin to see the neuromuscular junction is formed in a petri dish, revealed by the alpha-bungrotoxin. I think one of the most beautiful experiments that Hennig did is when he xenotransplanted this ES-derived motor neuron into the chicken embryo, and he can see that the mouse ES-derived motor neuron extending the axons outside of the spinal cord and innervating into the different muscle type of chicken embryos. So taking all of these criteria together, we believe these ES-derived motor neurons reflect the bona fide motor neuron identity in the embryos. So with this approach in hand, now we can begin to study many questions that hasn't been tackled before. For example, then we can begin to use this approach to recapitulate the process of motor neuron development. So my lab is interested in how non-coding RNA involved in this process. So we can begin to collect the cells from each stage during ES differentiation into motor neuron and do the transcriptome study. And as we can also get the motor neuron disease patients, fibroblast, and to reprogram them using Yamanaka factors and to generate the ALS iPS cells. So we can differentiate them to become motor neurons and to see whether the motor neuron degeneration process could also be recapitulated in the Petri dish. So as I just mentioned, so then we can collect the transcriptome, and we simultaneously differentiate them into spinal interneurons as a control, so can simply by adjusting the Sony Hedgehog concentration. So we collected cells from each stage and perform strain-specific RNA-seq. The reason we perform this is because many non-coding RNA don't have poly-A, such as circular RNA or enhancer RNA. So using traditional poly-A enriched technique may lose the information for those non-coding RNA species. And we simultaneously also perform small RNA-seq that allow us to get some novel microRNA as well as uncover some non-template modification of microRNA in different cell types. So this is a collaboration project with Dr. Hongrui Hong in National Jiao Tong University in Taiwan. So by systematical approach, we uncover several microRNA and long non-coding RNA signature genes in motor neurons. So by compare the transcriptome of spinal interneuron and motor neurons, we can systematically uncover around 40 microRNA that are highly enriched in post-mitotic motor neurons. And many of them are coming from two clusters. One is MN-ncRNAs cluster. The other is imprinting cluster. And today I'll just focus to talk about MN-ncRNAs cluster. So MN-ncRNAs cluster is one of the famous Uncle Mir in cancer field. In many cancers, MN-ncRNAs can be upregulated. But the function of MN-ncRNAs during neuron development and degeneration is not clear. So we first generated a conditional MN-ncRNAs motor neuron knockout mouse and to examine for the phenotype. So in this, in the middle of the figure, what you can see is to compare the control and knockout embryos. Different colors reflect the different motor neuron subtypes. So I think you can appreciate that in the thoracic region, we don't see much difference of the subtype. Yet from the lumbar segment, then we see a large portion of the red color of motor neuron is reduced in the knockout embryo. And this is verified by the secondary marker called ILDH2. And those these regions reflected by the FOXP1 and ILDH2 are the limb-innervating motor neurons that control the movement. And the embryos are not lethal. So in the movie you are going to see is that the embryo of the knockout or MN-ncRNAs in the motor neuron region is smaller. So in this video you are going to see this mice is smaller, although still very cute, but he or she is not able to move compared to the little mate. So this is consistent with that. Only the motor neuron control the limb movements are undergoing degeneration upon the conditional knockout. So that's interesting. But the question is, what is the target of MN-ncRNAs in motor neurons? So we tackle these questions by a systematic approach. First, we close the control and the MN-ncRNAs knockout embryos to a motor neuron reporter mouse line called HB9-GFP. So that allows us to isolate the motor neuron from the embryo. So we can get the control and knockout motor neurons and perform a microarray study. And then we can begin to look at those genes that are upregulated in the MN-ncRNAs knockout, showing in the green circle, as well as to intersect with the genes that are predicted to be the targets by MN-ncRNAs using target scan software. So we narrowed down that at least two family in the at least two microRNA in this family target to the genes as well as the upregulated genes, then we can narrow down to 18 genes that are mostly not reported before. But one caught our attention is Pten, as Pten was one of the most prominent targets by MN-ncRNAs in cancer field. And it's also positively regulate apoptosis that could account for our phenotype. But the function of Pten in motor neuron is almost entirely unknown. So we decided to use Pten as a proof of principle targets to verify whether it's a real target in embryo. So we did the independent qPCR experiment and verify that Pten indeed goes up in the MN-17 knockout embryos. Then we did immunostaining between the control heterozygous and the homozygous embryo. So if you look at the blue circle to blow up, then you can see in the FOXP1 limb-innervating mono at this stage, right before the apoptosis, they are still quite normal, so we don't see much difference. Now, if we look at Pten in the control, then you can see the Pten is known to be in the cytoplasm, control the PI-P3 kinase pathway and mTOR pathway. So its level is low, so there's no apoptosis. But if we look at Pten in the knockout embryos, we begin to see some upregulation of Pten in certain certain motor neurons showing by the red colors. But what draws our attention is that Pten now not only goes up, but also translocated into nucleus. And that's bizarre because we know microRNA control the target expression, but it doesn't control target subcellular localization. So that draws our attention. It might control other gene that can mediate Pten goes into the nucleus. So we would like to further tackle how does this happen, and secondly, whether nuclear Pten has any particular prominent function for apoptosis phenotype we are seeing. So when we went back to look at the literature, so it turns out Pten per se doesn't have NL as signal, so it cannot go into the nucleus by itself. But Pten can be mono-ubiquitinated by two E3 ligase called NEDD4 and ND51. So NEF4 and ND51 can mono-ubiquitinate Pten, and when the Pten is mono-ubiquitinated, it goes into the nucleus, and that will cause cell death and some chromosome integrity problem. And this is a beautiful study by Pandolfi's lab. Yet the Pten and the mono-ubiquitinated Pten goes into the nucleus, and the phenotype we are seeing in motor neuron hasn't been uncovered before. So we are wondering maybe MN-1792 can target to Pten, but also to E3 ligase. In this case, when MN-1792 is gone, all of the targets, including Pten, NEF4, ND51, all goes up. So that will lead to the mono-ubiquitination of Pten and goes into the nucleus. So to verify this hypothesis, we first check if the Pten and other E3 ligase level are affected. It turns out not only the RNA level is changed, but also the protein level. Here I would like to point out that traditionally using the embryos, you wouldn't be able to do Western blot due to the limited amount of motor neuron. So in this case, we use ESC derived motor neuron approach, so we can get ample resource of the motor neuron from wild type and MN-1792 knockout. So we can do the Western blot and some biochemical analysis data you will see. So you can appreciate in this Western blot, Pten, ND51, NEF42 are all upregulated at protein level. So that really suggesting they might be the real targets. But the goes up doesn't mean that MIR-1792 target to them directly. So we use a very standard ASIC, which is a 3' UTR luciferase. So we clone Pten, NDFIB1, F42, 3' UTR into the luciferase construct, and either overexpression in the wild type or 1792 knockout cells. Then compare the luciferase, and you can see with the negative control C6 that doesn't have 1792 target site. There's no difference between the knockout and wild type. But two known targets, ALEC2, Pten, this shows upregulation of luciferase activity using knockout when compared to the wild type. And NDFIB1 and F42 also show some upregulation. And this upregulation is rendered if we mutated the MIR-1792 binding site on the NDFIB1 and F42. So that's suggesting the E3 ligase complex components are bona fide MIR-1792 targets. So next, we would like to ask whether the MIR-1792 control Pten mono-ubiquitination and nuclear localization in vivo. So to study these questions, we overexpression MIR-1792 and the exogenous GFP Pten construct into the 293 cells. And in this construct, 1792 couldn't target to the exo Pten because it doesn't have 3' UTR. So whether the degree of the mono-ubiquitination of Pten will be reflected upon the E3 ligase degree. So in this case, that if we use IP to pull down exo Pten, you can see with the overexpression and 1792 together with Pten, we see the reduction of the mono-ubiquitination. And furthermore, if we settle subcellular fractionation of the cytosol and nucleus from the wild type and MIR-1792 knockout embryonic body, and you are going to see that Pten level is reduced in the knockout embryos. So that sort of suggests that the MIR-1792 controls Pten mono-ubiquitination as well as the nuclear localization. Then in the end, what we wanted to do is to see if Pten is really a prominent target to control this phenotype. Can we rescue motor neuron apoptosis by attenuating the Pten expression in the MIR-1792 knockout embryos? So to study these questions, we use conditional MIR-1792 knockout mice and further cross to the Pten heterozygous mice. So in this figure, you can see the green color reflect the limb-innervating motor neurons. In the knockout, it is going down due to the apoptosis. But when we knock down Pten in these knockout embryos, and I think you can appreciate that the number of motor neuron is spare, so suggesting Pten does regulate this pathway. So taken everything together, we suggest that MIR-1792 we uncover from the small RNA-seq is a limb-innervating mononeuron enriched microRNA. It controls the survival of limb-innervating motor neuron by targeting to Pten and the E3 ligase. So that makes Pten stays as a basal level in the cytoplasm and doesn't cause any apoptosis problem. But when we knock out MIR-1792, then we begin to see the level of Pten, E3 ligase goes up because of they are the direct targets of MIR-1792. And the expression level goes up with these three things together, leading to Pten undergoing a dramatic mono-ubiquitination and goes into the nucleus and to cause the limb-innervating motor neuron undergoing a specific apoptosis process. So we published this study in Cell Reports about 2 years ago, and this is a beautiful picture drawn by my mom that to reflect that we're using the mouse ear cells to decipher the motor neuron development and degeneration process and selected as a cover. And this project is pretty much contributed by two talented people in my lab. Jun-An Chen is a postdoc in my lab who pretty much conducted the project, and Yaling helped for many of the detailed knockout analysis and pioneer RNA-seq experiment, who is now studying in WashU St. Louis. So from this study, we believe that MIR-1792 might be an important regulator for limb-innervating mononeuron survival. And the reason this is the case is we know that during development, many neurons undergo apoptosis because they need to navigate, the axons need to navigate to innervate targets to get the neurotrophic support. But compared to the axial mononeuron you are seeing here, limb-innervating mononeuron travel for a remarkable long journey to innervate the muscles to get the neurotrophic support. So we think 1792 sort of provide a second layer of the battery for the limb-innervating mononeurons when during the navigation process they have fuel to make them not to become degenerate. So that recall us some of the phenotype of the motor neuron degenerative disease as ALS. The reason we think they could be not only a developmental important regulator, but also for the ALS is because we know amyotrophic lateral sclerosis as a neurodegenerative disease that only motor neurons are undergoing the selective degeneration. In the States, it's usually referred as Lou Gehrig disease because of the famous Yankees player, and some famous patients around the world were being recognized. And this disease being recognized in these 2 years due to the Ice Bucket Challenge. And this is one of the most nasty disease, as the patients lost the ability to move, but their consciousness are still very nicely maintained. And so in this disease, so far, there's no cure, and there's about $2 billion being spent on this subject. In the States, about $250,000 need to be paid by the insurance company of the family to take care of the ALS patients. Every 90 minutes, we know someone is diagnosed or somebody passed away due to ALS. Until now, there's only one drug called Riluzole can extend the survival only for several months with strong side effect. And as I mentioned, there's no cure. So there's still a lot we need to do to overcome this disease. And 2 months ago, a second drug that has been approved in the States, but the efficacy of this still needs to be evaluated in the following years. So we still need to find a way why the ALS only attack motor neurons and why only 10% and most of the cases are sporadic, and what is the reason for this. So to tackle this, we think maybe MIR-1792 could be an important factor. So Jun-An Chen, who conducted the project of the development of MIR-1792, further assembled a team with a very talented student, Guan Zhi, and medical Dr. R.A. Yanzhong in my lab to see whether MIR-1792 could contribute to motor neuron vulnerability in ALS and might serve as a target for the drug development. So we decided to tackle these questions by using SOD1. The reason is because SOD1 are still accounted for a large portion of the familial ALS disease, and we are currently testing whether 1792 is applied for other mutation as well as sporadic patients. So using SOD1 is because we have a very nice mouse model. So SOD1 G93A has been a working horse in this field and has very predictable phenotype. And when the mice are born at the beginning, they look very normal. Around 100 days to 120 days, we begin to see some onset of paralysis. And around 150 to 160 days, all of the animals will pass away. So using these mice, we first look at whether miR1792 microRNA is still expressed in a spinal motor neuron in adults, and if it is, if they have a motor neuron enriched pattern. So in this case, you can see by doing in situ hybridization together with immunostaining, chat refract the motor neuron region, and I think you can appreciate that miR17 is enriched in the ventral horn in the motor neurons. And with some quantification, we believe that miR17 has higher specific enrichment in motor neuron compared to the dorsal interneuron as well as some premotor neuron co-cal binding or rancher cells. So next, we would like to see if this is quantitatively, sorry, there's some delay on my side. Okay, yeah, so then in this case, you can see, so Guanzhi further using laser capture to isolate the ventral horn and the dorsal horn of the spinal cord, and to do the qRT-PCR of the family of microRNA in this cluster. And what he can see is most of this microRNA are enriched in motor neuron when compared to the non-motor neurons. So next, we would like to see in the disease content using SOD1, do we see any dysregulation of miR1792 as well as their target Pten and E3 ligase. So we profile the mice from 40 days to 80 days to 100 days. That is before the onset of the paralysis phenotype in the SOD1. As expected, we don't see the motor neuron change. Motor neuron number slightly reduced in 100 days in the SOD1 content. Yet, if we look at the miR1792, then we begin to see some interesting pattern, because in the control, miR1792 is still very robustly expressed in the control motor neuron from 40 to 100 days. Yet, in the SOD1 G93A mice, we see a robust reduction of miR17 at 80 days already. At P100 days, that motor neuron just begin to degenerate, we can already see a robust reduction of miR17, suggesting miR17 downregulation happens before the onset of motor neuron degeneration. So then we can next look at the downstream targets. How about Pten and other E3 ligase? So to look at this, we can see at using the laser capture experiment again to capture the motor neuron from the control and the SOD1 G93A mice. And when compare the SOD1 to the control mice, we see many of these microRNA clusters are significantly downregulated already at P100 days. And the downstream targets, including Pten, NDFIP1, NET42, are upregulated. And this is for the control that motor neuron, when we laser capture, the numbers are pretty much the same. So this is not a reflection of the reduction of the motor neurons. So taking this together, then we can now going to see what happened to the Pten and whether Pten goes into the nucleus as what we saw in the knockout embryos during development. So using this, we can take in the control and the SOD1 and perform very similar studies. And you can see, similar to the embryos, Pten stays in the cytoplasm in the control embryos. Yet, when we look at the SOD1 mice, I think you can begin to see some of the Pten goes into the nucleus and becomes quite prominent at P100. So if I segregated the channel and blow up, you can see in the SOD1 mice, many Pten goes into the nucleus. So taking everything together, let's suggest in the SOD1 mice, before the onset, miR1792 goes down together with the downstream genes goes up, and that's leading to Pten undergo mono-ubiquitination and goes into the nucleus. So this seems to be quite specific to motor neuron, because when we look at the dorsal interneuron or the premotor neuron Kell-binding rancher cells, there doesn't seem to be a Pten enrichment and MIR-1792 downregulation in the SOD1. And that is consistent with the SOD1 ALS mice, only motor neuron undergo degeneration. So with this data together with the previous motor neuron data, we believe MIR-1792 downregulation in motor neuron seems to be a signature pathway, and this might provide some explanation why motor neurons are selectively undergo degeneration in ALS mice. Yet, as I mentioned at the beginning, in this disease, most motor neurons undergo degeneration, but some motor neurons don't undergo degeneration. For example, the motor neuron controlling the eye movement, ocular motor neurons, are resistant. But there are also some motor neurons undergo particular early degeneration. For example, the axial motor neuron controlling the axial muscle and the limb-innervating motor neuron controlling the limbs. It seems that the yellow colors of the limb-innervating motor neuron show the earliest degeneration. But it is still unknown why this is the case. But if you recall the phenotype I mentioned at the beginning of my talk, the MIR-1792 knockout embryo seems to have a specific limb-innervating motor neuron degeneration. So we are wondering maybe the particular vulnerability of the limb-innervating motor neuron in the ALS is due to the MIR-1792. Yet, this is a difficult question to tackle in vivo because most of the limb-innervating motor neuron and axial motor neuron markers disappear in the adult motor neuron. So there's no good markers to do that. So to tackle this, to overcome or to circumvent this hurdle, we decided to go back to use the embryonic stem cell approach, that we can establish a new reporter, allow us to delineate the different motor neuron subtype. In this case, we give the FOXP1 GFP from Jeremy Dayson, and we made the HB9 RFP transgenic embryo, and by crossing them together, then that allows us now to delineate the two different subtypes, which is, you see here, which is the limb-innervating motor neuron showing in the yellow colors and the axial motor neuron showing in the red colors. And those are just the markers to verify their authenticity. So don't worry about the detail of the names. So now we can cross this transgenic embryo back to the control and SOD1 G93A mice, derive their ES cells, then differentiate them to become motor neuron. And we can fact sorting the two subtypes, either the limb-innervating motor neuron or the axial motor neuron, which one show in yellow, one show in red. And you can see the markers are very nicely segregated for the two different populations. So suggesting this approach can really get the subtype from the disease and the control. Yet, they are still embryonic motor neurons, so we need to make them to become mature and to let them to undergo degeneration process. So we use a defined medium to make them to become mature and use ER stressor. This is a collaboration with Sebastian Thames in Karolinska Institute and Henning Whitley in Columbia University. So we can use these stressors to see whether there's any subtype difference of the degeneration degrees upon aging and degeneration. So it turns out that the axial motor neuron MMC upon the stressor doesn't seem to affect too much of the survival. Yet the limb-innervating motor neuron LMC, after 24 to 30 hours, then we begin to see a large reduction of the motor neurons. And after 36 hours, when compared to control, we see a large degree of apoptosis. That suggests to us that the limb-innervating motor neuron does undergo early degeneration. So next, we can begin to look at the MIR-1792, Pten, and every hallmark I just mentioned at the beginning. In the axial motor neuron MMC, we don't see much difference. Yet, in the limb-innervating motor neurons, then we see a significant downregulation of miR-1792, concomitant with the upregulation of Pten, anti-FIP1, NEF42. Those are the targets of 1792 I mentioned and in the first part of my talk. So that suggests the subtype vulnerability. Now, so in the next slide, I'm going to show when we use Pten to do the staining. And in the axial motor neurons, then we don't see Pten goes into nucleus, similar to what we showed before. It stays in the cytoplasm. Yet, when we look at the limb-innervating motor neurons, then in the control set, we do see many of them are still in the cytoplasm. Yet in the SOD1, we begin to see the accumulation of the nuclear Pten. And those are quite repeatable with several batch of the experiment, and the quantification is shown in the right. So taking everything together with the in vivo mouse model, that we show you that miR-1792 Pten, nuclear Pten, as a hallmark of the motor neuron degeneration. But in this model, we wouldn't be able to tell you the subtype difference. With the embryonic stem cell approach, now we can tell you that in the limb-innervating motor neurons, we see more strong change of miR-1792 as well as the nuclear Pten. So together with the in vivo and the in vitro approach, we suggest miR-1792 regulates the degeneration of the limb-innervating motor neuron in the SOD1 mice. But I think every one of you will be curious whether 1792 Pten only serves as a good marker, or are they actually doing anything if we manipulate the label and to see the phenotype? And this is exactly what Guanzhi further tackles. He is wondering to what degree that miR-1792 overexpression can ameliorate the ALS symptom and extend the lifespan in SOD1. So we first use the genetic approach. I know it's a very complicated genetic approach, but don't worry. So it's just 3 different colors represented 3 different genetics. The control as without miR-1792 ALS, red color, no 1792 but not turn on. With the green color is the motor neuron specific turn on genetic approach to make 1792 overexpression in the ALS. So in this case, you can see in these 3 sets of the spinal cord, miR-17 is downregulated in the ALS, as we showed before. With this genetic overexpression approach, you can see miR-17 is now back to normal, pretty similar to the wild type, with not to exactly the same level, but pretty much to the normal type. And this is the chat marker, so you can see it's in the motor neurons. Then we now can compare the phenotype of these 3 embryos. First, we look at the Pten expression. And again, with the ALS in the middle panel, I think you can see many of the Pten goes into the nucleus. And in the rescue overexpression 1792 mice, you can see many of the nucleus Pten now is back to the cytoplasm. And if we compare to the motor neuron numbers, we can see not only Pten level is reduced, but also with the increase of the motor neuron numbers. So how about the morphology? So we know this mice has a hallmark of kyphosis due to the muscle weakness. So in the middle panel, you can see ALS mice with the kyphosis on the back and with the weakness of the limb. But when we overexpression on miR-1792, we can see the kyphosis phenotype is largely rescue. And we can also see that in the hind limb, the weakness is also restored. So suggesting morphologically, they look okay. And how about their motor behavior, motor ability? So we perform standard rota-rod experiment, as you can see in this. So we train them and to see how good they can perform on the wheel. And so this is the behavior analysis, you can see in the ALS mice, in the red colors, they begin to show reduction of the motor ability when the disease onset at 100 days. Yet when we overexpression with the MIR-1792, we can see the ability is pretty much restored to normal until 130 days and begin to drop a little bit. So it seems to delay the onset, but eventually the mice will still die. But this is also, SOD1 is one of the most notorious strain to do the rescue. So we are testing now in other strain to see whether this can has as good function as we see in SOD1. And this is a movie to show at 130 days the paralysis of the hind limb. So they don't move, but with the overexpression, they seem quite happy. So taken everything together, this pretty much suggests that 1792 not only serve as a good marker, but can be a therapeutically application to delay the onset. Finally, we look at the survival. So that's about 20 to 25 days increase of the lifespan when we overexpression, and this is actually quite good considering rescue in the SOD1 G93A mice. So with all of the information I talked to you today, so we use embryonic stem cell as an approach together with the mouse genetic paradigm, and we uncover MIR-1792 as one of the important microRNA regulator to control limb-innervating motor neuron during development, and does so by target to the Pten and E3 ligase to control the subcellular localization of Pten. And during aging process for ALS patients, MIR-1792 seems to downregulate it. And if we, and that account for the limb motor neuron vulnerability for the ALS patients. And when we overexpression MIR-1792, then we can see that the Pten will be restored into the cytoplasm, and the motor neuron number can be rescued, and all of the motor behaviors and the lifespan seems to be ameliorated as well. And so we believe this could be a good target for future development of the ALS drug therapy. So I think in the future it would be interesting to see whether this could be a good candidate for gene therapy, or in the other familial ALS or sporadic ALS phenotype, and we can see the same effect. So I introduced you two projects that is pretty much conducted by my postdoc Jun-An Chen, and she is leading the team of Guanzhi, Yanzhong, and Yaling to perform this project. And we have several collaboration internationally and locally in Taiwan. And so I appreciate for your listening, and I'm happy to take any questions. Yep.
And Jun-An, thank you very much for an extremely interesting, thought-provoking presentation. So I'd just like to remind our audience, our attendees, to please go ahead and submit any questions you would have for Dr. Chen in the QA box on the lower left part of the screen. Jun-An, so with that, we do have a couple of questions that are loaded up. Maybe we can start with those.
Okay.
So for our attendees, I will, so I'm your moderator, will go ahead and read the questions so everyone can hear them, and then give Dr. Chen an opportunity to answer them. So in no particular order, Dr. Chen, so maybe we can start with, so why do you think the MIR-1792 plays, I guess, a fairly specific role in a subset of innervating neurons, and not, more broadly across, more of them, I suppose?
Wow, that's certainly an interesting question. So we are now, so in the field, we know limb-innervating motor neurons are particularly vulnerable to degeneration. So we also know the ocular motor neurons are resistant. So Chris Henderson did a very beautiful study to show that NMP9 account for this differential vulnerability. And so what we provide here is another one that could be a modifier for this. And we also know that there's some resistant differential degree of alpha and gamma motor neurons. At this moment, we still don't know whether there's a different MIR1792 accounts for the alpha and gamma motor neurons. So we are currently tackling this question at this moment. But I think that will be one of those. I think we are stepping into the precision medicine eras, and so we think that each drug might have a different cure ability for the different subtypes. So looking at subtype, I think, is certainly one of the directions to tackle in the future. Yeah.
Okay, thank you. And okay, here's another, I guess, somewhat related question. So have you looked at the impact of soluble neurotrophic factors such as GDNF in the regulation of MIR1792?
Yeah, so that's interesting. So at the beginning, so we think that MIR1792 is an intrinsic microRNA to regulate the limb-innervating mono survival. So in the field, I think we believe that the neurotrophic factors control the survival. There's no doubt about it. So it's purely rely on the extrinsic neurotrophic factors, or it's more the intrinsic. So we did an experiment as right before aiding a neurotrophic, and we fact sorting. And since that the neurotrophic factor doesn't affect the dying of the MIR1792 derived motor neuron. So we believe that MIR1792 is the intrinsic, independent of the BDNF or GDNF control. Yet we also would like to be cautious that this kind of experiment need to be done more systematically in vivo. So we couldn't completely rule out, but by in vitro we think it's more intrinsic instead of the neurotrophic factor reduced regulated microRNA. Yeah.
Interesting. And I guess maybe in part you answered, but I guess is an overlapping question, and that would be, do you think that that glial cells may play, or other stromal or supporting cell types play a role in the regulation of MIR1792 in this kind of pro-survival function?
Yeah. So that's a great question. So glial cells' role in the neuron degeneration have been increasingly recognized in the field, and some of the beautiful study by Sergei Popovsky as well as Kevin Egan, they suggest that the astrocytes can accelerate the degeneration of the motor neurons. So we haven't tested that, but simply by overexpression MIR1792 in motor neurons by genetic approach seems to increase the survival. So I think cell autonomous function of MIR1792 is definitely recognized. Yes, we are using a Cre driver that also can express the 1792 in the oligodendrocyte. So it could be that the glial cell might also contribute as well. And so we are currently pursuing with motor neuron specific Cre to drive MIR1792 by viral delivery, and to see whether by simply increasing 1792 in motor neuron is sufficient enough. So if that is not the case, then we will try the glial together with motor neuron and see if it's actually help with more therapeutic effect. Yep.
Great. Very, very interesting. So I have, actually, this is a question from your moderator, but I'm curious if you think there for the other, as you described early in your talk, hundreds of different subtypes of motor neurons involved in innervation into a wide array of muscles obviously throughout your body, and many of these are broadly affected in the neurodegenerative diseases. I mean, do you think that there are other classes or other if potentially you would set screens up for different subtypes of motor neurons, do you think you may tease out other non-coding or microRNAs that may have roles aside, parallel or similar roles to 1792, but maybe different for the different neural subtypes?
Yeah, so we haven't looked at very carefully. So we started with the 1792 and look at the motor neurons. So the difficult part at this moment is to get a large amount of different subtypes. For example, with Hennick's approach, it's quite straightforward to get the brachial motor neuron, but getting the lumbar or thoracic motor neuron is relatively difficult. So a lot of people are trying to find a good way to get more the different subtypes, but it's still not very robust at this moment. So we haven't looked at other motor neuron subtype and to see maybe other microRNA or other non-coding or parallel or RNA that might be more enriched in those population, and thereby accounting for maybe why some of them are resistant, some of them are more vulnerable. So I think with the ESL differentiation approach, although it's very powerful, but it does have some limitation. So I think we also need a mouse model or human IPS model as a complement to study all this topic at the same time. Yeah.
Right. Very, very interesting, and it shows the power and also maybe points to some of the current limitations of these systems and models too.
Yes.
I think given the time, maybe one last question for Dr. Chen, and this is a little bit of a open-ended or broader question. So we'll toss this your way. But that is, as you, particularly, your in vivo and rescue studies or overexpression studies in the SOD knockout, it looked very promising as potential therapeutic or as alleviating the disease and maintaining longevity. So I guess I ask you, is this in your view? What would you see as the challenges for mRNA-based therapeutics for this type, for delivery and use in humans in motor neurons? I mean, do you see this as a viable route? And I guess if what would be the major hurdles? And you have 3 minutes or less to answer that. No, just kidding.
Yeah, that's certainly a $1 million question, right? So I think it's promising, but has, as you say, tons of the hurdles need to overcome. But I think one optimistic thing is that FDA just approved the gene therapy, and so the gene therapy now is back to the stage again. So by reason 2 papers by NEJM to suggest that the SCAAV9 SMN1 delivered into the SMA seems to work quite well at the early phase. So we believe that with the SC or AAV mediated, the adeno-associated virus mediated approach for gene therapy might provide an opportunity in the future. So that's having said that, I recognize that many hurdles need to overcome. So I believe a cocktail approach in the future, either with the small molecule or with the the gene therapy, depending on the patient's situation, might provide a more rational therapy in the future. By simply applying one particular molecule or one particular gene may not be able to do all of the job for many cases of the sporadic ALS patients. That's my point of view.
Well, they're very, very thoughtful. And so, Dr. Chen, thank you very much for your presentation and questions. A fascinating talk, great, really exciting work. Thanks for sharing it. And and again, thanks to our attendees. I hope we hope that you enjoyed the session. And once again, thanks, Jun-An. It was a terrific talk. We really appreciate it.
Thank you very much. Thank you, David.
Our pleasure. See you.